So today we're going to be showing you how to swab Chinook salmon samples, or any other fish really, for the mucus for getting DNA. Here is a swab, a sterile swab that will be used for the mucus grabbing. So here you can see that it is right now in its packaging, and how you want to open this packaging is at the metal hook side. You'll open it up, and you do this before you get your fish in hand, and that's because it can be difficult to open it while you're also dealing with a fish. So make sure not to touch that that particular swab part, and that's because that is your sterile surface that you only want to come into contact with the mucus. And if it touches anything else before that, just go ahead and throw that away. It's totally fine. All right, so once your swab is ready and you've made sure that it is nice and sterile, if you ever touch anything with that tip, you can just get a new one. That's totally fine. How you're not going to do it is by very lightly just rubbing the surface, not moving that tip around very much, the swab tip. Instead, you want to make sure, when you're doing it properly, to be moving the swab around, and so what you do is kind of a twirling action of that swab to try to really maximize how much mucus you're getting. Simultaneously to twirling, you are applying firm pressure, not so much that you're actually going to be hurting the fish, but it is firm enough to be able to grab that mucus off of the surface of the fish so that we can really get as much as possible. Then you open up your tube of 1X phosphate buffered saline, or PBS, and now you're going to start mixing it. And you mix it vigorously for about 20 seconds or so. And the purpose of this is to try to get as much of that material, the fish material, which contains mucus, it may contain scales, off of the swab and into the solution. So after the mixing is done, then what you're going to do is you're going to start rimming around, trying to get as much of that liquid out of the swab as possible. And the purpose of that is really just to ensure that we are maximizing our total material that we can get.